this is your first tour and first exposure of one of our DCCs. And what's great is the reason we want to use this one as our example is we've really used Metabolomics Workbench as an example for the Knowledge Center also of how do we make some of the results accessible to someone who may not even know what metabolomics is and may want to use it in the research. So you can see this is a theme that's going to come through the rest of the session. So with that, I will turn it over to Mano and Shankar to take the calm.
Thank you, Noel. Hello, everyone. We are very happy to provide a very, very quick hitchhiker's guide into the Metabolomics Workbench. This journey started about 20 years ago when we got a large grant called the Lipid Maps. It's a GLUE grant. And at that point in time, the last time when anything with nomenclature or organization of metabolites, especially lipids, was done was in 1960s. So it was kind of, all over the place, and mass spec was just coming into picture. And then came the next 10 years, which really mushroomed into large measurements of metabolites, going from hundreds at a time to tens of thousands of metabolites. And this launched the initial project. NIH, recognizing the importance of metabolites, started in the early 2000s. A project, a common fund project, which involved the National Metabolomics Data Repository and the Metabolomics Workbench, along with funding 10 other groups for generating data, vast amounts of data. And this journey now is continuing with funding from Metabolomics Workbench from the Common Funds, Data Commons Workbench from the NIH CFDE Phase 1 project, and the CFDE Data Resource Center. I wear two hats. I wear the hat of the co-PI along with Dr. Avi Mayan, for the Data Resource Center, and I've been the PI for the Metabolomics Workbench for 12 years at this point in time. So a lot of people have contributed over a period of time. I'm not going to acknowledge every one of them. I'll just going to only introduce Mano, who will then talk about the next 15 minutes about Metabolomics Workbench. Let me walk you through why metabolomics in a few seconds. We know the central paradigm in biology. DNA gives RNA gives proteins, but then proteins act on metabolites. Metabolites are ubiquitous in human physiology and in mammalian physiology and other physiology. And these are functional endpoints of physiology and pathophysiology. They are kind of reasonable scale of analytical challenge because they are small molecules to medium-sized molecules. They are measurable from serum and blood, which is fantastically important because they're easily accessible body fluids. And then they are a direct mirror of environmental influences, malnutrition, like diet, exercise, medication. And so it really is an endpoint. And we can quantitatively metabotype individuals, just like genotyping. We can characterize the dynamic state of an individual through metabotyping. What are the challenges in metabolomics? Well, how do you measure metabolites? Today, mass spec can produce tens of thousands of metabolites in one single measurement. How quantitative is metabolomics? With standards now, it's become extremely quantitative. So we can longitudinally monitor a human being for their metabolites in blood or serum, and we can look at the changes. How can we analyze the omics measurements to obtain metabolite information? We have been developing tools the community has been developing tools for the last 2 decades towards mining these omics metabolomics measurements. How can we organize, store, and use metabolomics data? I would like to say that National Metabolomics Data Repository, which is NMDR, and the EBI metabolites are the two unique resources in the world which essentially provide organized, store, and provide metabolomics data. How can metabolomics data be integrated to provide a systems view of biology? This is the quest of jointly Data Resource Center, Knowledge Center, as you will hear later on, and of course, Metabolomics Workbench. What are the implications for systems medicine? Can we metabotype an individual? Please wait for the future. This is something we are actively engaged in terms of thinking about a rubber stamp of a dynamic state of a human being. What NIH decided to introduce this in the Common Fund program. They wanted training in metabolomics. They wanted comprehensive metabolomics resource course, metabolomics technology development, data sharing and international collaboration, and reference and synthesis standards. This is what Metabolomics Workbench has led to. I'm going to now turn it over to my colleague, Dr. Maurya, who will go through the next, 10 minutes, the next few slides. Mano, over to you.
Thank you, Shankar. So I'm going to provide some details about what the Metabolomics Workbench resources are. So starting with this slide, here we get a glimpse of different assets that we have in Metabolomics Workbench, for example, we have over more than 3,000 studies deposited, out of which more than 2,700 have been made public. Similarly, many of them, more than 1,000, are related to human studies, and we have more than 3 lakhs samples, and we have some other details provided here, which I'm not going to cover in complete detail. So let's go to next slide. So this slide provides an overview of how, starting with the data and metadata deposition into the system, how we channel it through different processing steps, and then to various tools, and how it is visualized on the Metabolomics Workbench website. So starting with the experimental data that is generated by different data metabolomics researchers, we have an online submission system where we have the ability to provide named metabolites, which and all that information, along with the other metadata information, is deposited into a relational database. Next, please. So once this data and metadata is in the database, we have a series of tools as part of a comprehensive toolbox, which can be applied to these different studies, and we can do different types of analysis, some of which I will cover a little bit later. In the case of untargeted data, the metadata part goes into the regular database that we have, and the detailed untargeted data goes into a different sort of file-based system. Next, please. Because it's untargeted data, in this case, M over G values are not being able to map to a particular metabolite, so it goes through a different pipeline, and eventually it is also channelized to a different set of tools. And again, at least some of that data can be visualized in various means. Next slide. So this slide provides an overview of how the data deposited into NMDR has grown over time. As we can see here, it has continuously been growing, and particularly in December 2015, an automated online deposition system was introduced based on all the experiences that we gathered over the previous 3 years, and we saw a substantial increase in the number of studies deposited after that, and the momentum has continued. Next slide, please. So in the Metabolomics Workbench, in different sections, we can look at the data based on different aspects. For example, we can browse the studies based on the species name, for example, whether it is from human, mouse, or rat, and we can see those 3 are the top represented species across all the studies. Similarly, we can look at it based on the disease, for example, cancer and diabetes, and then obesity. Those are 3 are the top three disease that we have seen in the samples in the studies deposited. Next slide. Similarly, there are other ways to sort of look at, slice the data and then see them and further follow through. For example, based on the sample type or the type of human pathways that are covered by the metabolites that have been measured in these studies that we can see on the right-hand side. Next slide, please. So I will briefly go over the capabilities of Metabolomics Workbench. For example, we can submit metabolomics data and then query and search across the studies based on certain keywords corresponding to different types of attributes, and then be able to download the data for further custom analysis. And then, of course, use various tools on the website itself, for example, for visualization, analyzing the data, and other types of integrative analysis. Next slide, please. So before I go into the actual examples of studies and analysis, different analysis tools, we wanted to point that we store much of the data, much of the metadata in a format called MW tab, which essentially corresponds to Metabolomics Workbench tabular format, which has various sections such as metadata section, data section, and some additional information can be provided as comment. And a view of that is provided, a file view of that arrangement is provided on the right-hand side. In the metadata section, we have information such as the project, study, experimental variables, referred to as factors or different parameters that define a specific condition under which that for which that sample corresponds to, and other related information. In the data, we have the actual data values and tables. Next slide, please. So now going to the actual different parts of the website, the most prominent one from a user perspective is to browsing and searching through the different studies, where we can browse them and search based on different aspects, and also use REST services to fetch the data from data and metadata from Metabolomics Workbench. Next, please. So, for example, when searching for the studies, this table provides a overview where we have the ability to sort them based on different columns like study ID or study title or species or analysis and so on. And then we can select a particular study and follow on that further. In this table, we also directly provide a way to download much of the data related to that study. Next slide, please. Besides MW tab, another very important feature of NMDR is that we use a reference metabolite system called as RefMet, which provides harmonization and standardization of metabolite names. And that's very important for being able to integrate the data from different studies or across other resources. It's a unifying nomenclature. It provides the ability to perform comparative analysis across different studies. Next, please. In reference system, for many of the metabolites, exact structure has been also linked to a metabolite structure database. And then a comprehensive classification system is provided based on various labels of details, which is used for data visualization and statistical analysis. And all this facilitates integration with biochemical pathway tools that helps us map these metabolites to different further interpretation and annotation of these metabolites. Next slide, please. So going to various tools that are available for the study, we can select a particular study, and within a study, there could be different measurement modes, for example, GCMS, positive ion mode, negative ion mode, and so on. And once we choose that, we have a set of tools listed on the respective pages where we can click choose any one of those and then select the parameters related to those and then carry out the analysis. What is shown on the right-hand side is just a summary view of the results from different types of analysis, which some of which we are going to look at with respect to specific studies. For example, looking at study number ST001140, which is related to changes in the canine plasma lipidome after short and long-term excess glucocorticoid exposure. Okay. So what is shown here as a summary view is different types of analysis. For example, volcanoplot analysis that we see on the right-hand side, the left bottom panel. And then we can also do class enrichment analysis as shown on the right-hand side. Next slide. Maybe I can pause while we address the questions.
Yeah, there's two questions in the chat. One about cumulative statistics. I think the question is, what is the URL for this cumulative statistic page, or what are the instructions for selecting it from the Metabolomics Workbench homepage?
So I can address that. We have not shared that URL, but because largely that has been for internal use to gather that statistics. But I believe we should be able to provide that link. I believe the part of the reason it was not provided directly on the website is that in order to compute those tables, it requires a little bit of computation. I mean, searching through all the databases. But we can certainly look into, as long as it doesn't put too much pressure on our infrastructure, it should not be a problem.
Right. All right. I think we're good. Keep on going.
All right. There was another question. Yeah, certainly we can update that.
You can access it without logging in.
Oh, okay. Thank you. Okay. So looking at some of the tools for analyzing this data for study 1140, for example, we can do clustered correlation analysis, which we discuss on the next slide. So here we have some parameters, for example, what is the cutoff we want to choose for correlation, and which MS analysis we want to include in our study. Or I believe we also have the option to include all of them to get all the metabolites included. So what this shows is a correlation heat map for that data set, which kind of provides an idea which metabolite groups are more correlated to each other and so on, which we can further analyze. Another example is network analysis on correlated metabolites, corresponding to mapping it to classification and also mapping it to fold changes. Next slide, please. So this slide essentially shows a tool provided by a Michigan group. And actually, this also kind of shows that we have the ability to, at least to some extent, incorporate compatible tools developed by other groups into the workbench. In this case, given the data matrix for metabolomics data, we have the ability to generate networks based on what is known as a debiased sparse partial correlation. And we can look at this network further based on various filtering and so on. Here is another example of building network based on a mixomic package. This one is non-interactive, but I believe we do have the ability to select different cutoffs for the correlation so that we can get more dense or sparser network here. And for all these results, we provide a way to download the results with it's a PDF file or in some other format. Then we have an extensive set of tools, or extensive set of means to extract the data from Metabolomics Workbench via the REST services. And this slide provides just a little bit a glimpse into that. The actual web page provides a lot more descriptions of how to construct the REST URL, including the example given here. Next slide, please.
Mano, we can wrap up quickly in the next 3 minutes, maybe.
Right, yeah. So besides the core Metabolomics Workbench resources, as part of the CFDE effort, we have been developing some cool tools. For example, what is shown here is called MetGene, which provides a workflow where starting with a gene or a set of genes, one can find what are the reactions and metabolites associated with those genes. And in Metabolomics Workbench, what are the studies where those metabolites have been measured and related information. Next slide, please. Another tool that we have developed is called MetNP, which is another workflow which lets one either analyze the data from one of the studies as Metabolomics Workbench, or the user can upload their own data in a compatible format, and we find out what are the differentially altered metabolite between two conditions, what are the pathways or classes to which they map, and it generates a network for the metabolites and the different pathways and various other results. Many of these tools are actually available through as Jupyter notebooks as well as shown here on our GitHub page link. Next slide, please. So now I think, Shankar, you can take over.
Thank you, Mano. So I'll wrap up in just 1 minute. Beyond metabolomics, we want to really derive biological knowledge in deep ways. And so this is already, Christy showed you the slide. I mean, Common Fund Data Ecosystem entered phase 2 with introducing DRC, Data Resource Center, Knowledge Center, Quick CW, Cloud Implementation Center, and Center for Training, and a Coordination Center linking all of those. And so we are now entering into a phase where we want to marry the DCC in an integral way with everything else that is going on in CFDE. In fact, we have this diagram which was shown before. And we also provide, in response to Hunter's earlier question, we really provide a link to external repositories and resources such as, all of us, STEADY, TopMed, EPIC, breast cancer projects, and dbGaP, etc. And so, in fact, this is one of our goals in DRC to really bring in synthesis of all of these in a queryable way. And the biomedical research community, I mentioned to you about the Knowledge Center. The Knowledge Center is the key towards the next phase of building knowledge from all of these collective resources. And I will turn it over now to Noel to continue with the presentation.